My hope is that my computer works so much faster off my iTunes library, which took up like half of my hard drive. So I'm not restarting my computer before every lecture now. Okay, so we're going to talk about glycolysis, and eventually we'll get to wine and beer, but not sadly. So metabolic pathways. So we kind of did this overview of metabolic pathways, the kinds of pathways that we're going to see. So glycolysis is definitely of the linear variety. So we're going to see substrate going into the pathway, 10 enzymatic reactions, and spitting out the final product at the end. Citric acid cycle we're going to see as a cyclic pathway, and then fatty acid metabolism, both fatty acid breakdown and fatty acid biosynthesis are spiral pathways. We're not going to talk about them yet, but glycolysis is a linear pathway. So why would we want a metabolic pathway with distinct enzymatic steps? So if we're going to, we're going to talk about burning fuel or oxidizing fuel. Okay, so we could just, so we already learned that cellulose, so plant matter, stores glucose, so homopolymers of glucose in cellulose. We could burn cellulose, and we're going to oxidize the glucose, right? Would that work to oxidize glucose for a fuel source for us? So it's still oxidizing glucose, but we're not really going to get the kind of energy out that we want, right? So what kind of energy would we get out if we burned, so if this is burning fuel. So how would the energy be released if we set sugar on fire, for example? Heat and probably light, okay? So that's probably not going to help us metabolically, right? We're going to do the same oxidation, but now chemical oxidation. We're going to end up with the same products. So if we have C6H12O6 glucose, at the end we're going to have carbon dioxide and water, but the products are going to be different. So instead of heat and light, we're going to end up with ATP and reduced cofactors. And those we can actually use. Okay, so these discrete steps not only give us energy in a form that we can use chemically, but we can control them. Okay, so we don't necessarily want to light all of our sugar on fire at once. We want to go through in smaller energy release steps to get not only the chemical form that's useful, but in a way that we can control and regulate those enzymatic pathways, or the enzymatic steps in the pathway. We've already hinted at this a little bit. So where have we seen feedback inhibition of an enzyme before? Okay, what enzyme did CTP feedback inhibit? Close. ATCase. Very good. Should get a point for that, right? ATCase, aspartate transcarbamylase. I promise not to say anything important in the last 5 minutes. How's that? So aspartate transcarbamylase, remember, is the enzyme that's the first enzyme in the biosynthesis of pyrimidines. So it's the committed enzyme, and CTP is the ultimate downstream product, and so it comes back and feedback inhibits the first enzyme in the pathway. So you could say that the product here is CTP, and this enzyme E1 is ATCase. Okay, it's going to feedback and dial down the activity of that enzyme so that we don't make something we don't need. Okay, what about feed-forward activation? What would that, can you imagine why you would want to do that? Okay, amplification. What do you mean by that? So signaling, yeah, we'll get to signaling, amplification of signal, but I wouldn't necessarily call it that. Okay, so to keep the metabolic pathway going, okay, so it's almost like product B is giving enzyme E4 a heads up that there's something coming downstream. Okay, so you're kind of priming the pump or giving it a heads up that, hey, you're going to have to dial up your activity because even though B isn't a substrate, B will lead to C, will lead to D. Now, if D is a substrate, you want to have that enzyme ready to go at its full capacity to go on and continue the pathway. So we'll see an example of that. Certainly, feedback inhibition is more commonly used than feed-forward activation. So you kind of have to look for examples of feed-forward activation. There's a whole lot more of inhibition. Okay, so the major catabolic pathways that we're going to take glances at in metabolism. So if we start with sugar, so these are based on all the nutrients that we consume. So proteins, sugar, and fat. Okay, and remember we said that proteins are not typically an energy source. They're more often used just in recycling to make more proteins. But certainly they can feed into different steps in metabolic pathways. Okay, but we're going to emphasize this a little bit right here for today and maybe Tuesday. And then ultimately we'll get to citric acid cycle. We'll touch on fatty acid catabolism just a little bit at the end of the semester. But sugar metabolism is a great example of all the ways that enzymes can be regulated and some of the mechanisms. Okay, so we talked a little bit about thermodynamics and bioenergetics. So those are the problems that you want to practice from the end of the book problems. So anytime that we have calculations, those are the ones that the more you practice, the easier it's going to be on an exam. Okay, so my exam, MCAT, ACS standardized exam, you should practice, so kind of like Henderson-Hasselbalch, Michaelis-Menten, the more you do, the easier it is when you see it. So if we think about the thermodynamics of metabolism, we want to think about now, are we at equilibrium in an enzymatic reaction, or are we at steady state? And so I'll give you the hint that in metabolic pathways, we don't tend to be at equilibrium, we tend to be at a steady state. So for any one enzyme, so equilibrium would mean, our forward reaction and our forward reaction and our back reaction kind of have evened out, and so nothing is happening. So at equilibrium, remember, there's no energetic driving force to go one way or the other. Okay, so wherever we are in terms of concentrations, we've reached, hard to say a definition without using the word, so we've reached a state where the reaction isn't going forwards or backwards, we're there, we're at equilibrium. In metabolic pathways, however, we could have, so we would call this flux. So if we have a certain amount of A, we're going to keep going through the pathway until we get to C or whatever the downstream product is. And so we want to not build up a whole lot of B. So however much B we make, we're going to go on to make C from that. So it's not like there's no B there, but there's a steady state amount of B. So that would mean that whatever this rate is, ideally, the rates are going to be equal. Okay, so whatever's going in the first enzyme is also going to go on to the second enzyme so that we don't have a buildup of one of the intermediates, because that would be pretty inefficient. So it's almost like you have a bucket of water and you're equalizing the flow going in with the drip going out the bottom of the hole. Okay, so you don't want the bucket to overflow and have this buildup of B that you're not really doing anything with, you're not getting the chemical energy out of, nor do you want to drain the bucket, so you have to wait around to make more B before you can go on to make C. Does that kind of make sense? Yes? So the absolute amount isn't so relevant, it's just that whatever absolute amount there is isn't going up and down a whole lot. So yeah, it's hard to say. So in a cell, there would be a certain amount of any metabolite under certain metabolic conditions, and it might be millimolar, it might be micromolar. It just kind of depends on the just steady state levels of that metabolite. So for some, even if you just look at all the amino acids, there's different concentrations of each amino acid. They're all, probably in the low millimolar range, 1, 2, 3 millimolar, but they're not all exactly equal. And they're certainly going to be more than some other, metabolite that might be micromolar. So the absolute amount's not so critical. And so remember, we also talked about how most metabolic enzymes, how would we describe them? And we talked about 2 kinds of metabolic enzymes. I'm not coming up with clues for you. So most metabolic enzymes catalyze reactions that are near equilibrium. Okay, so that means, so even though we say that the enzyme itself is not at equilibrium, they're close, which means you have a little bit more A, now it's going to shift the equilibrium to make B. Okay, if you have If B increases for some reason, then you're going to shift the reaction to make A. Okay, so that just means that an enzyme can react to the concentration of metabolite that's there and drive the reaction forward or backward. So most metabolic enzymes catalyze near equilibrium reactions, but the pathway overall is steady state. Does that help, or did that confuse the matter? Just file it away. File it away. We'll come back to it. Question?
Is the steady state on B only applies if the A is like infinite or keeps on? Is there so much?
So is B steady state only if there's an infinite amount of A? Let's not think about infinite amounts of A. And so for many of these metabolic pathways, so again, we could pick one of the intermediates. So we could pick an intermediate of glycolysis. It's probably, so its concentration, it's going to fluctuate kind of in this range. It's not going to go way up or way down.
Yeah.
Okay. If enzymes so for the enzymes in the pathway that are near equilibrium enzymes, that means that if A builds up a little bit, that's going to increase the flow through that pathway. So again, B is not going to change very much. So it kind of depends whether you're talking about the control points in the metabolic pathway, which we'll get to, or the enzymes that are near equilibrium. So kind of the center reactions of a metabolic pathway, those concentrations of intermediates are not going to change very much. They're going to respond to the influx of substrate at the beginning, but like B is not going to change very much. A even is not going to change very much. So if you think of, again, we're going to talk about glycolysis, so A then would be glucose. So think about what situation where glucose might increase significantly. After you eat, right? So you have a meal, your blood glucose goes up, and your body wants to not keep it at that condition, but do something about it. So now some of the glucose is going to go into glycolysis, right? Some of the glucose, what would happen to it? Okay, so taken up by cells in the liver either to do glycolysis or what? Stored as glycogen, right? If you really have a surplus and your body can't adjust to it, what might you do with that glucose? Say it again. Okay, you might make fats. And if you have uncontrolled diabetes and you can't do any of the above, what are you going to do with it? You're going to urinate it out. You're going to have high sugar in your urine as a clue that, whoa, we're trying to get rid of all the sugar. It's not going anywhere the places it's supposed to go. We just have to get rid of it. Okay? So if A is glucose, it's going to fluctuate, but then your body wants to deal with it one way or the other. And then B, so the next metabolite in the pathway, it's not going to change very much because you're only going to put glucose into that pathway if it can be used downstream. Okay? So let's start talking about glycolysis. So glycolysis is the set of 10 enzymes that will process sugar as a fuel. So carbohydrate, sugar metabolism. We'll talk about how it's also fed by amino acid and lipid pathways, but we're going to ignore that for now. We're just going to say we're starting with glucose going downstream. And so glycolysis happens in every cell type. And for some cells, it's the only way that cell can get energy. Okay, so the brain has to do glycolysis. So the brain can't get ATP energy any other way except taking sugar and processing it by glycolysis. There's a little bit of an exception to that, but that's only under cases of panic starvation mode. Okay, so if you're on a desert island, you've used up all your glycogen stores, you're not eating, where's your body going to go for energy? So it's going to degrade your muscle, and it's going to burn fat, and there are byproducts of fat metabolism that your brain can use as kind of a last ditch. I'm going to try to keep this sucker alive for another couple of hours, and that's all we're going to do. Okay, but for the most part, under normal, healthy conditions, the brain has to have sugar. And glycolysis is pretty efficient. Okay, so what can glucose do? So we kind of already talked about this. So we can store it, we can burn it. We'll see kind of a side pathway. The pentose phosphate pathway is used if we need to make, so what do you think ribose 5-phosphate might be used for? So RNA, ultimately for DNA. So for reduced cofactors, so if you have a surplus of sugar, you can also enter this pentose phosphate pathway to make nucleic acids. So under what circumstances would you want to make a lot of RNA or DNA? Or maybe what kinds of cells make a lot of RNA and DNA? Skin, hair, so rapidly dividing cells. Skin, hair, lining of your epithelium, so your stomach lining. If you're pregnant, you're making a lot of cells. What other not so great scenario? Cancer. Okay, so cancer, pregnancy, you're going to do a lot of nucleic acid biosynthesis. Also, this is the pathway that makes reduced cofactors for fat biosynthesis. So if you have a surplus of sugar and you don't need it because you're just kind of sitting, maybe you break your leg, you probably also need it for healing. But if you're just sitting around eating bonbons and not exercising, but eating a lot of sugar, ultimately the reduced cofactors will make fat. So that's the sugar turning into fat pathway. And then also, whatever structural polymers we need to make. That's where glucose goes. Okay, so we're going to dig into glycolysis. So we're going to go through these 10 enzymatic steps. It's not as scary as it seems at first. So how many of you have seen glycolysis enzymes in cell bio before? Okay, so hopefully some of the names will sound familiar. Maybe you haven't drawn the substrates before, but you will. Okay, so make some flashcards this weekend. And so the net reaction of glycolysis is if we start with a molecule of glucose, we're going to end up with 2 molecules of pyruvate. Okay, and we're going to make some ATP. We're going to reduce NAD plus to NADH. So remember, if we're burning the sugar as a fuel, so we're oxidizing the sugar, then we're going to have to reduce something. Okay, so there are redox enzymes involved in glycolysis. So if pyruvate is the oxidized version of glucose, then NADH is the reduced form of NAD+. So I guess I should say reduced form. So NAD+ gets reduced into NADH. Glucose gets oxidized and cleaved apart into 2 molecules of pyruvate. Okay, so those are the 10 steps. They look messy, but they're not really. You'll remember them, I promise. If there's one thing that Wake Forest pre-meds are good at, it's memorizing. So this is one time where you got to memorize. Okay, but hopefully doing the mechanisms will also help you understand the rationale, and so that it's not such a big deal about memorizing them. Okay, so there are two phases of glycolysis. So the first is the hexose stage. 2 stages or phases. Hexose stage, that just means we have 6 carbons attached to each other, right? So we're starting with hexoses. We'll go through each of those separately. We're going to end up with trioses. So these are 3 carbons, right? So if we start with a 6-carbon sugar, we're going to cleave it eventually into 3 carbon molecules, ultimately getting to pyruvate. So if those slides are useful to kind of have all the information in one place, that's great, but now we're going to go through them separately. And again, so we can also call the 2 halves of glycolysis, so we can call them the hexose stage and the triose stage. We can call it the, so we're kind of making investment here and payoff here. Okay, so we're going to have to use some ATP in the investment or the hexose stage, and then we're going to get ATP out in the payoff stage, so the net payoff of glycolysis is 2 ATP molecules. Not really that big a deal, right? So 2 ATP molecules, big deal. What's more important, though, is the reduced cofactors that we're going to get. Okay, so 2 NADHs. Any idea how many ATP molecules we get ultimately from each of those NADH molecules? It's like 2.5, isn't it? Like 2.5? 2.5 ATP per NADH. 2.5 or 1.5? More than 2. Okay, 2.5. Yeah, so it used to be that everyone wanted to think of them in whole numbers. We don't have to be limited by whole numbers anymore. 2.5. I meant to refresh my memory on that, but it's 2.5. Okay, so, big deal. We get 2 ATP molecules. In the payoff, we actually get 5 more from the NADH. Okay? And then we're going to get from pyruvate in the citric acid cycle, we're going to get even more reduced cofactors. So ultimately, we're going to get a lot of energy out of just that one glucose molecule. Okay, so to bring us back to enzymology, so remember we learned the different kinds of enzymes that there are, the 6 classes of enzymes. So of the 10 enzymes of glycolysis, we're going to see lots of transferases. So we're going to see 4 kinase reactions. Those are all transferases. We're going to see 3 isomerases. We're just going to move things around within the molecule. 1 dehydrogenase. So dehydrogenases are class 1, so there's oxidoreductases. And I kind of hinted at this last week. Dehydrogenase should automatically make you think NADH. Okay, so dehydrogenases are hydride transfer reactions. So hydride transfer to NAD plus or hydride transfer from NADH to make NAD plus. And then 2 lyase reactions, so making, breaking double bonds. Okay, and we're going to see today, if we get to it, 2 mechanisms. More than 2 mechanisms. We're going to see lots of mechanisms. Okay, we're going to see a kinase. I already did hexokinase, an isomerase, and maybe we'll get to the dehydrogenase as well. Okay, so first reaction of glycolysis is hexokinase. So we know, as soon as we see kinase, we know that it's a transferase, and we also know that ATP is going to be involved. Okay, so we're just going from glucose to glucose 6-phosphate. I did this mechanism when we were talking about ATP coupled reactions, remember? So I would much rather draw glucose as a linear molecule. Okay, so if that's glucose, so if we're going to go from glucose to glucose 6-phosphate, remember the hydroxyl group on the C6 is going to be the nucleophile to attack the alpha phosphate of ATP, right? Sorry, gamma phosphate. I need a vacation day, right? P-O-P-O. You know what? Some enzymes actually bind ATP like this, honestly. Okay, so if the hydroxyl on the C6 is the nucleophile, remember it's going to attack the gamma phosphate, open that up, ultimately cleave that phosphoanhydride bond to give us glucose 6-phosphate. So you can draw it in the ring form, or you can draw it in the linear form. Like that. Okay? Easy peasy. Okay, so you already know how to do one mechanism. Yes?
Did you do or show like a mechanism where the alpha phosphate is attacked?
So that was the aminoacyl tRNA synthetase.
Okay.
So the amino acid will attack the alpha phosphate of ATP, cleave off the beta gamma phosphates, the pyrophosphate, so that you have an amino acid adenylate, so an aminoacyl AMP. So again, the purpose of that is to activate that carbonyl carbon. So yes, we did that mechanism. Okay, so you're just plucking off that gamma phosphate to phosphorylate that glucose. And so why is that favorable? So we see a negative standard state delta G. So why is that so negative? Yes? Okay, so we're hydrolyzing ATP. So almost any time we hydrolyze ATP, we're going to have a negative delta G. Okay, so that's going to be a favorable reaction.
Is that thing that you draw on the right top?
Mm-hmm.
Is it just like the linear form?
Yeah, it's just the linear form. Yeah. You can draw the cyclic form of glucose and glucose 6-phosphate. Ultimately, we're going to work with the linear form because it can't stay cyclized for very long.
Okay.
So On an exam, you can start with glucose ring form or linear form. I just like to draw linear form. For some reason, your textbook likes to draw it in ring form. So it can stay in the ring form for the first reaction, but ultimately it's going to have, so of course that's anomeric carbon, so that's a reducing carbon, right? That's the reducing end, so that's going to be in equilibrium. Okay, so glucose is going to be in equilibrium with its linear form. Glucose 6-phosphate is going to be in equilibrium with its linear form. Ultimately, it's not going to cyclize anymore. Oh, and there's the mechanism. Yep. So that's just a prettier version than my scribbles. So that's one you'll need to know. But again, it's easy. You should be able to do that already. Okay, and then phosphohexose isomerase. Okay, second reaction. We're going to start with glucose 6-phosphate. And so it's an isomerase, so we're just doing an internal rearrangement. But we're changing it from glucose to fructose. And if you'll notice, is that significantly favorable? No, but it's near equilibrium. Okay, so if it's at equilibrium, what would the delta G be? 0. Okay, so I think the rule of thumb is if it's kind of less than, so if the absolute value of delta G is less than or equal to about, 6 kilojoules per mole, that's still near equilibrium. Okay, so if it's like 32, positive 32, not so near equilibrium. If it's negative 30, it's definitely thermodynamically downhill. Anything in the 1, 2, 3 kilojoules per mole, you can imagine it going backwards too. Okay, and in fact, this isomerase can go both ways. Very much a near equilibrium reaction. Okay, so if we draw them in their linear forms, so glucose, as you know, is a aldohexose. Okay, so we have an aldehyde group up there. Fructose is a ketohexose. So how did that double bond move? So how do we interconvert an aldose and a ketose? I wouldn't exactly call it a tautomer. Not exactly. Yes, Katie. Okay, yep, so it's definitely an isomerase. And we're going to see acid-base chemistry in the isomerase. Okay, so what we're going to do is we're going to have to deprotonate. So this is the mechanism. And so even though this base on the enzyme is written as just kind of a generic base, it turns out that in most organisms, this phosphohexose isomerase uses a glutamic acid. So the glutamic acid is going to deprotonate that proton on the C2 to make this double bond. So that intermediate is called a cis-enediol. Okay, so cis-enediol because you have double bond there. And both hydroxyl groups are on the same side of the double bond. Okay, so enediol means carbon-carbon double bond, 2 hydroxyl groups. That's an enediol. Cis-enediol is where both hydroxyl groups are on the same side of the double bond. Okay, so then it's just a reverse of that. Okay, so if we go from the cis-enediol, now we just need some other base to come in and deprotonate that hydroxyl group. Now again, we're going to make the double bond in a new place, and that the electrons in that double bond are going to open up and grab the proton back from that original general base. Okay? Yes?
So that H that's on the top alcohol, that's just from like acid that's in there?
Yes. So you're saying up here? Yeah, so it's just going to pick up, yeah, so that would be that one. So it's just going to pick up so you could say, if that makes you nervous, you could say there's some general acid there. Okay, so that general acid will donate its proton, and then later, so maybe now it would be nicer to say this is the deprotonated general acid. So this is general acid-base chemistry. So what was the general base in the first step is going to be the general acid in the second step. What was the general acid in the first step is going to be the general base in the second step. So sometimes you don't even really specify what those are. You just say, yeah, there's proton shuffling. Okay, but it's certainly easier to learn it if you think kind of specifically about base deprotonation and acid donating a proton, and then everything just kind of shifts back the other way. Yes?
And that proton from the glutamate takes is the most acidic. That's why it leaves.
Over here, you mean?
Step 1. So,
Oh, you mean so that this proton is most acidic? Yes. Yep. Well, and it's probably also how the enzyme holds the substrate. So it has to be acidic enough, and proximity effect. So that's the one that's close to the glutamate to take it. Yep. And so that probably means that whatever that general acid is, it's also positioned properly then to be the proton donor. But again, this is a very near equilibrium reaction. So you could also imagine that the enzyme could hold the product and just do everything backwards. Okay, so now we have this isomerase, and we've ended up with fructose 6-phosphate. So we've gone from glucose 6-phosphate to fructose 6-phosphate. Don't bother drawing the rings for me. Okay, just keep it in a linear form, but it could equilibrate with the ring form. Does that make sense? Yes. Okay, so I'll give you a little bit of foreshadowing. So of the 10 enzymes of glycolysis, 7 of them are near equilibrium, which means they can go forward in glycolysis, or they can go backwards in what pathway? Gluconeogenesis. Okay, so hexose phosphate isomerase is one of the ones that's near equilibrium. It's in both pathways. And I'll give you another hint. For the ones that are very negative delta G, they're not near equilibrium. Okay, so hexokinase is not in gluconeogenesis.
Are we learning the mechanism for all?
Nope. Most of them, though. Well, and there's 4 kinases. Once you know one, you know them all. Yep. And there's 2 isomerases. They're a little bit different, but not that different. Actually, 2 or 3 isomerases. I think 3. Okay, so here's another kinase. So we have fructose 6-phosphate. Here's the second step where we need to consume a molecule of ATP. Same deal, we're going to grab the gamma phosphate from a molecule of ATP, and now this time put it on the 1 position. Okay, so now we're going to have fructose 1,6-bisphosphate. Again, a negative delta G standard state. So again, favorable, going in the forward direction. Mechanism is the same, just using a different hydroxyl group. And it's important, so this is not just called phosphofructokinase, it's called phosphofructokinase 1. Doesn't seem like a big deal now, but that 1 will come back to haunt you. Okay, so phosphofructokinase 1, it's named partly because I think that was the first one to be discovered. More importantly, however, the phosphate group gets put on the 1 carbon. So remember, phosphofructokinase 1, the phosphate gets put on the hydroxyl on the carbon 1 of fructose. Later, we're going to see PFK2. Where do you think the phosphate goes on that one? The 2nd carbon. Okay? So it's not an enzyme of glycolysis, but it's an enzyme that helps regulate glycolysis. You're so smart. Okay, so now the next, this is the enzyme that kind of bridges between the hexose stage and the triose stage, because aldolase is actually going to cut fructose into 2 trioses. Okay, so we're going to go from fructose 1,6-bisphosphate, and if you think about where the phosphate groups are on the linear form of fructose, they're on the 2 ends. So now if we cut the carbon-carbon bond between the C3 and C4, now we have 2 3-carbon molecules and a phosphate group on each end. Very convenient. So I will tell you, so why do we phosphorylate these sugar molecules? Any idea what phosphorylating them has to do with anything? So one help that phosphorylation gives us is that phosphorylated sugars don't cross the cell membrane. Okay, so when glucose comes into the cell and gets phosphorylated, we don't have transporters to take phosphorylated sugars out of the cell, so it's kind of like it's trapped there. So it's not going to leave until it does what it has to do. Okay, and so that's just going to help with, that kind of helps with regulation, so we're not going to lose those intermediates. They're going to stay where they are until they've made it all the way through the pathway. Okay, so now aldolase is going to break the bond between the C3 and C4. And so these 2 names, they might seem kind of weird. I think we've talked about these molecules before. So we've certainly talked about glyceraldehyde. So why did we talk about glyceraldehyde? Do you remember? So that's the simplest sugar that dictates the D and L configurations. Remember that? So glyceraldehyde. So it's chiral. So glyceraldehyde has only one chiral center, right? So that's chiral. But now if we put a phosphate group on there, so now it's glyceraldehyde 3-phosphate. And so again, if it helps you to know that, to remember the structure of glycerol, great. If you don't already know the structure of glycerol, don't bother memorizing it. That's not something you need to know. It's just that if it's already in your memory banks, it might help you with the naming. And remember, what's acetone? Okay, so that's acetone. Remember the stuff you wash the glassware with in organic chemistry lab? So dihydroxyacetone would be that. Okay, so if we have hydroxyl groups on the two ends, so that's dihydroxyacetone, and now if we slap a phosphate on one of the positions, now you have dihydroxyacetone phosphate. So again, you don't need to remember the kind of organic molecules unless it helps you in memorizing those two molecules. Not memorizing, learning, remembering. Okay, don't panic. I will not make you learn the aldolase mechanism. Okay, so it's a little bit messier. We're not even really going to go through with it, except the features I want you to remember are that it's an enzyme that uses covalent catalysis. So I don't know if you can see right there, there's a lysine residue in the enzyme active site that will attack the C2 of fructose 1,6-bisphosphates, and so we end up with this covalently attached substrate. So aldolase uses covalent catalysis, and the name of this intermediate, so if we have an R and double bond C, so this is called a Schiff base. So that's all I want you to remember about aldolase. So it uses a Schiff base intermediate, and it uses covalent catalysis. So basically the enzyme uses that lysine residue to kind of hold everything in place, and then we're going to have bond cleavage to release glyceraldehyde 3-phosphate. Then we're going to have some more acid-base chemistry to hydrolyze off the dihydroxyacetone phosphate. So the enzyme is going to hold it in place so that chemistry can happen to the 6-carbon molecule, cleave off the first 3 carbons, and then release the last 3 carbons. Okay? We're going to break that carbon-carbon bond. Yes?
The Schiff base, is that just like what that is called, or like what happens is called a Schiff base?
So that intermediate, so that N double bond C is called a Schiff base. That's all I want you to remember. It's not something you see terribly often in enzymatic reactions, and so it's just something to remember. It's just a little detail. Okay, so if we think about where the carbons go, so it's almost like if you have your 6-carbon 1,6 fructose 1,6-bisphosphates, we're going to almost fold the enzyme in half. Fold that 6-carbon sugar in half. So we're going to cleave there, and now we're going to have the phosphates on the 1 and the 6, but now this glyceraldehyde 3-phosphate, if you kind of flip it upside down, it's going to look kind of like that dihydroxyacetone phosphate. Does that kind of make sense? So aldolase is going to cut the carbon-carbon bond between 3 and 4, but now we need to have an isomerase to interconvert those molecules for the rest of glycolysis. Now we're going to proceed after that with 2 3-carbon molecules, which we'll see in a minute. Okay, so the dihydroxyacetone phosphate is going to get converted to glyceraldehyde 3-phosphate, and the glyceraldehyde 3-phosphate will continue. Okay? But you can kind of keep track of the accounting of the carbons. So the 3-carbon on glyceraldehyde 3-phosphate could have come from the 6-carbon or the 1-carbon of fructose 1,6-bisphosphate. So again, it's like that 6-carbon molecule folds in half to become 2 3-carbon molecules. Okay, we'll see that in the subsequent steps. I will let you guys take a minute break if you want. You're starting to glaze over a little.
You just want that to happen.
Mm-hmm.
That you just roll that dihydroic acetone and turn it into glycerol.
Right, so we'll see. So we have another isomerase.
It's more useful.
Well, because is it more useful? So it conserves the enzymes.
Okay.
You know, so now we don't need. So for the last however many steps, we don't need two different enzymes. You know, so you basically add an enzyme to interconvert them, and then you proceed with two molecules of the same thing.
Okay.
You know, so the glyceraldehyde 3-phosphate that came from the fructose 1,6-bisphosphate, it can continue, and then you just kind of add in one more enzyme to like get the other guy in the same path.
Okay.
We gotta learn how to change it from the dihydroxide.
Yep, that's the next enzyme.
Claire and I wanted to ask that teacher if we could come see the school. So could
Oh,
I
yeah.
have your email address?
Yes,
Or is it on the school's website?
probably not
'Cause she's
because she's so
not
new.
teaching yet.
Yeah, I will forward it to you.
Okay. Thank
Yeah, definitely. That's great. Send me an email to remind me, Katie. Yes, it's in my office and your exam. Yep. How was your trip?
It was really cool. It was really cool.
When did you get back? Monday? Sunday?
Tuesday night.
Oh, so you're still like, you're awake. You must be like,
About 5:30 in the morning.
Yeah.
So right now, have my coffee.
good. You're doing well. You don't want to get up and do some jumping jacks or anything?
I was up till like 4 in the morning, just wide awake. I'm like, well, I guess I can go to sleep now.
Yeah. You should stand up and stretch at least. Mike, did your hair turn that golden spontaneously? So you didn't put anything in it? You didn't put lemon juice in or anything? Okay, not to accuse you, but that looks like too quick. I don't know. My brother-in-law was like blonde, you know, so he spent summers at the Jersey Shore. And looking at him now, you'd never guess that his hair was so blonde in the summers because it just went from brown to like very blonde very quickly.
So go away, coming back to North Carolina.
Yeah.
So I finally.
So in the summer I used to put, so in the 80s there was something called Sun In, and it was basically lemon juice. You spray lemon juice on your hair and you get a very nice, so yeah, it's not very good for your hair. Go to the beach, have like highlights. Kids today. I want to tell you that we sprayed ourselves with baby oil too, but that wasn't so healthy. Okay. Everybody have a good stretch. Okay, so the next enzyme in the pathway, okay, so if we've cleaved our fructose 1,6-bisphosphate, and so we have glyceraldehyde 3-phosphate, so it can go on to the rest of the steps, so the triose phase, triose payoff phase. The dihydroxyacetone just needs an additional enzyme to get it into glyceraldehyde 3-phosphate as well. And so this is what I was trying to say, that so the phosphate that starts on the 1 position of fructose 1,6-bisphosphate, when it gets isomerased, now that position is the 3 position of glyceraldehyde 3-phosphate. Okay, that still maybe doesn't make sense. But the 1 from glucose now becomes the 3 on glyceraldehyde 3-phosphate. It's almost like we just flip it upside down and renumber the carbons. Don't freak out by that too much. Okay. So if we start with dihydroxyacetone phosphate, now we're going to isomerize that to glyceraldehyde 3-phosphate. And again, you know, so maybe the standard state for energy change isn't quite in the range I said, but it's still a near equilibrium reaction. So it can go forwards or backwards. Okay, and so how does that work? Oop. Was I going to do that pathway? Maybe not. Okay, never mind. We're not going to do that. But what does it look like? So what mechanism that we already did does that look like? Yeah, hexosaisomerase. Yep, phosphohexosaisomerase. So practice that. I'm not going to do it, but see if you can turn this into an aldose. Okay, so just basically make the mechanism go backwards. So we can do that together on Tuesday if you want, but see if you can do that on your own. Okay, so now we have 2 molecules of glyceraldehyde 3-phosphate. So I'm not going to say 2 from now on. We're just going to follow one of them through, essentially. But now we're going to do even more oxidation to get to pyruvate. Okay, so here's the triose stage overview. We're going to go through each of those individually. So here's the oxidation step. So glyceraldehyde 3-phosphate dehydrogenase. So we're going to go from glyceraldehyde 3-phosphate. Ultimately, we're going to use a molecule of phosphate. But if we just think about putting it on, how on earth would that happen, right? So it's going to seem kind of odd. How would we get that phosphate group to that carbon? We're going to go through an oxidation step first and then put the phosphate group on. Okay, so again, the delta G is still near equilibrium, but now we're going to see our first enzymatic reaction involving a redox step. And again, it's a dehydrogenase. So what is going to get transferred? What form are the electrons going to get transferred? Hydride, so an H minus. Again, it kind of freaks you out to think of an H minus. That doesn't seem quite right. Okay, but H minus is a hydride. And so we're going to see some redox chemistry. So there is the mechanism, but it's hard to see there, so I'm going to do it. Well, did I leave a free slide? Yes, I did. Okay, so I'll do it on here so it's all together for you. Okay, so the enzyme has an active site cysteine residue, and we're going to start out with it deprotonated. So either that means its pKa is really different than you would expect, right? Or there's some general base that's going to come in and deprotonate it. So we're going to start with it already deprotonated, but just know that it's going to have an altered pKa from what you learned from cysteine normally. Okay, so we're going to draw the glyceraldehyde 3-phosphate kind of upside down, just to kind of put everything in the right place. Okay, so that's glyceraldehyde 3-phosphate, upside down. This is C1, C2, C3. Okay, and so what do you think that cysteine is going to do? So you've seen this, so you could almost even pretend that it's what kind of reaction? You can almost pretend it's a cysteine protease. Okay, so remember papain from the exam? So that cysteine, what's it going to do? Okay, so it's going to attack that aldehyde carbon, right? And that's going to open up. Okay, so now we have a tetrahedral carbon. And then what do you think is going to happen next? So it's certainly going to collapse. But now we need to bring in our friend NAD+. So this is the business end of NAD+. So the R would be this big old thing that's going to have a ribose group on it, an adenine. You don't need to know the structure of that. Okay, so I need you to know the business end of NAD+, NADH, but not the rest of the messy molecule. What's that? No, it's not a shift base. No. So if that, question? So if that oxyanion collapses, so don't think of this as a proton. So it's not an acid-base reaction. It's a redox reaction. So we're going to draw the arrow moving from those electrons. Sorry, I drew that a little far away, didn't So both electrons in that bond are going to leave. So that's the hydride, right? Okay, so a hydrogen and 2 electrons, that's the hydride. And so both electrons in those bond are going to leave, in that bond are going to leave. And it looked kind of messy there, didn't it? So if that's the R, here's the business end of NADH now. Okay, and in fact, let me draw that H in a different color just to be clever. Okay, so here's the H that came from here. So the H and the 2 electrons came from that C1 position of the glyceraldehyde 3-phosphate. So why do I draw 2 H's there if only one of them came from the substrate? One was there already, we just didn't draw it. You know, so in that ring, there was an H on all those positions, right? So here's where kind of drawing it the lazy way hopefully doesn't confuse you. But each of those positions on that, so it's not a benzene ring because there's a nitrogen in it, but in that ring, each one of those had a hydrogen on it already. But now we're going to move a hydride into that ring. Does that make sense? So one of those hydrogens and both electrons came from the glyceraldehyde 3-phosphate. So now this is NADH.
So that is a hydride.
Yep. Okay, so that was NAD plus. Then when the hydride is attached, when the hydride gets transferred, now those electrons move around the ring to sit on the nitrogen, so that now you have the lone pair back on the nitrogen. Does that make sense? Yes. So would it help if I did it on the board so that stays there, but you can see it a little bit more blown up? I like to have it. Yeah.
So is it good that
Yeah. So that's the only. So that's basically the redox center. Yep. That's where That's where it's gonna end up. Okay. So if we then have. Okay, so we could imagine that we would hydrolyze that product off with a molecule of water, but we don't. Question? Did I mess something up?
Yeah.
Oh, golly. Very good, thank you. How's that? Thanks. Okay, so now we have to get that product off, right? So that looks like what we want it to look like, except not quite. So instead of using a molecule of water as the nucleophile now, we're going to use a molecule of inorganic phosphate. So this is called a phosphorylasis. So instead of a hydrolysis, oops, not physics, Okay, so it still has the lysis, so it's still going to break a bond, it's going to cleave something off, but instead of hydrolysis, so a water molecule being used, it's phosphorylasis, so a phosphate molecule is being used. So that phosphate is going to attack that carbonyl carbon. We're going to do kind of shorthand here, so it's going to open up, and it's going to collapse, then it's going to cleave off. So that we're left with, and now don't freak out, but I'm going to draw it upside down again. I'm going to flip it around. Okay, so that plus, we're going to get the enzyme back. So again, this is the C1, 2, 3, C1, 2, 3. So now we're left with 1,3-bis-phosphoglycerate. So bis-phospho means there's 2 phosphate groups, but they're on different atoms. So it's not a diphosphate like ADP, it's a bisphosphate. So there's two phosphate groups but on different places. They're not connected to each other. So 1,3-bisphospho, instead of a glyceraldehyde, it's a glycerate. Okay, so we've now oxidized that carbon. Okay, so it's not an aldehyde, instead of a glyceraldehyde, it's a glycerate. 1,3-bisphosphoglycerate. Everybody okay with that mechanism?
Eight again on a name?
I mean, it's an O minus. It's a
Okay.
yeah. So glutamate is a carboxylate.
Okay.
Yeah, so it's a carboxylate. Yep. And so, basically that's a carboxylate with a phosphate group attached to it. So 1,3-bisphosphoglycerate. So glycerate means it's a carboxylate. Bisphospho means you have two phosphate groups. They're not attached to each other, but they're both attached to the same molecule. I know, too much nomenclature. So here's our 1,3-bisphosphoglycerate, looking a little prettier than my drawing. Okay, and now this next step of glycolysis is the first time we get paid back in some ATP. Okay, so this 1,3-bisphosphoglycerate is at a higher energy level than ATP. So cleaving off that phosphate is even more energetically favorable than cleaving off the gamma phosphate of ATP. So the kinase that we're going to see next, so usually we think kinase uses the molecule of ATP. So this kinase is named for the backwards reaction. So this kinase actually makes a molecule of ATP. Okay? So we're going to go so the phosphate group is going to get transferred from 1,3-bisphosphoglycerate. So from the 1 position, now it's just going to get transferred from 1,3-bisphosphoglycerate to a molecule of ADP to make ATP. So basically the ADP is going to come in and pluck off that phosphate so that now this phosphate becomes the gamma phosphate of the ATP molecule. Make sense? So it's basically named phosphate as if you're taking off that gamma phosphate, but really the reaction in glycolysis goes the other direction to make a molecule of ATP. So it's our first payback. Okay, and it's energetically favorable downhill. Okay, and so that's direct transfer. So we're generating ATP by direct transfer of that phosphate group. Everybody okay so far? Question? Okay, and so again, now we have another, so mutase, what class of enzyme do you think that is? Isomerase. So phosphoglycerate mutase is an isomerase. Okay, and so we're going to go from 3-phosphoglycerate to 2-phosphoglycerate. So the question is this phosphate there, do you think that's the same phosphate as that one? No. Very good. Okay, so we're going to actually use an enzyme that starts out phosphorylated. It's going to donate its phosphate group to 3-phosphoglycerate to make as an intermediate 2,3-bisphosphoglycerate, and then it's going to grab off the phosphate from the 3 position. Okay, and so think ahead. Where have we seen DPG before? Don't answer yet. Think about it. Okay, so the mechanism for this one, not too surprising either. So we will have done way too many enzymes in one lecture, but that's okay, because then you'll have plenty of time to study them this weekend, right? Okay, so phosphoglycerate mutase. So if we start with 3-phosphoglycerate, okay, so there's 3-phosphoglycerate, and we're going to, so the enzyme has 2 histidine residues. And one has a phosphate group on it, and one doesn't. But they're both in the active site of the enzyme. And again, you don't have to copy this. You can do it later if you want. Yes. Okay, so we have one that's going to act as the phosphate group donor, and one that's going to act as a general base. Okay, it's pretty obvious to tell which one is which, right? So the histidine on the bottom is going to act as a general base. And so what proton do you think it's going to take off? Do you think there are any acidic protons there at all? So do you think that? No. Sorry. Good guess. There's two. Okay, so this one is going to be the more acidic. Okay. You're just setting me up, so I look smart, right? So this is the general base. It's going to, long distance deprotonation. So it's going to deprotonate that hydroxyl group so that now that oxygen will grab that phosphate group. Okay, and now the electrons can go there. So now what do we have? So now we have 2,3-bisphosphoglycerate. And we have a histidine that's dephosphorylated. And one that's protonated. Okay, and so now what do you think is going to happen? So probably the one that's naked, so we have a naked histidine, right? But it has to end, we have to get back to where we started, right? So now this histidine is going to grab the other phosphate that started out on the molecule, and the oxygen then is going to grab that proton from that histidine. So no, that's probably too many steps at once. You can break those out into individual steps if you want, but really it's just an exchange. So the 3-phosphoglycerate becomes 2-phosphoglycerate through the intermediate 2,3-bisphosphoglycerate. So now we end up with 2-phosphoglycerate. Oops. Okay, so 2-phosphoglycerate. Question?
Yeah, what makes it want to grab that?
I don't know. So it's near equilibrium. So does 2,3-bisphosphoglycerate change the active site structure a little bit, maybe? And so now the intermediate kind of shifts subtly. So now the 3-phosphate is near that histidine. I don't know. Yeah, it's just what the enzyme does. Don't ask questions. Yeah, don't ask why. Don't ask enzymes why.
Should those be hydrogens on the...
Oh, sorry. Thank you. That one you mean? Just that one, or is there another one? Was that the one you meant, or a different one?
Yeah, sure.
I think that's right. We still have... Yep. Yeah, so that's our glycerate. Remember? That's our carboxylate. So our oxidized aldehyde, essentially. Okay, so that is that mutase mechanism. So remember, where have we seen 2,3-bisphosphoglycerate before? Hemoglobin. Okay, so before we kind of just called it BPG, but it's actually 2,3-BPG, 2,3-bisphosphoglycerate. Remember, it's very negatively charged. And so where does that BPG end up in hemoglobin? So it's in the core of that quaternary structure. What does it stabilize? The T state. Okay, so BPG stabilizes the T state of hemoglobin, which leads to offloading of oxygen. So what does oxygen binding or delivery have to do with glycolysis and sugar metabolism? So ultimately, as we make this pyruvate, we're going to feed it into the citric acid cycle. Then we're going to feed it into the electron transport chain in mitochondria for respiration. So ultimately, we're going to need oxygen available to the tissue. So it's kind of a, I don't want to say it's a feed-forward mechanism because I'm not sure that it really is, but it's kind of giving, kind of setting up respiration so that there's oxygen available. So as glycolysis is happening, you're making, so your transient levels of BPG rise a little bit. So now that's going to offload more oxygen from hemoglobin to kind of get you ready for respiration. So even though we said it's steady state, that little bump in BPG will help in overall respiration. So oxygen binding, it is correlated with glycolysis. Okay, so if the black line is normal, so hexokinase, what do you think if we have a hexokinase deficiency, are we going to make more BPG or less BPG? Less. Okay, so now at every oxygen concentration, we're going to hang on to the oxygen a little bit more. We're going to stabilize the R state. If we have, so we haven't gotten to pyruvate kinase yet, but just kind of imagine that, so hexokinase is enzyme 1, pyruvate kinase is enzyme 10 in the pathway. So if we have a deficiency in the last enzyme of the pathway, we might have a little bit of a buildup of BPG, okay, and so that's going to favor the T state. So at every oxygen concentration, we have less oxygen bound, so it's offloading better. So up here, you could say hanging on to oxygen. So it's not delivering it as well. So R state stabilized. And down here, we can say delivering O2 more, and it's favoring the T state. Okay, so glycolysis and oxygen binding, oxygen release are related to each other through that little molecule BPG. Crazy, I know. Okay, we are out of time. We are nearing the end of glycolysis, but you can certainly work on what we've done this weekend before Tuesday. No quiz, though. No quiz on glycolysis. And you can kind of give yourselves a heads up on the rest of it if you want to make those cards as well. Okay, I will see you Tuesday. Have a good